Hello, my name is Elaine Ficarra, and my STAR project looks to uncover SIV pathogen resistance in wild Cameroon chimpanzees. I've had the honor of working in Dr. Gonder's lab, supervised by Miss Dana Mitchell. And our motivation for this research is the biodiversity hotspot in mainland Africa. There are two subspecies of endemic chimpanzees in Cameroon that need conservation efforts. Moreover, understanding their genetic makeup and what drives their natural selection can help us to develop the best conservation initiatives. To do this, we furthered our research on how these chimpanzees are genetically different. And a possible process to explain their genetic diversification is pathogen resistance to the simian immunodeficiency virus, or SIV, which is the precursor to HIV. So insight from this research can be applied to humans as well. But turning the focus to Cameroon, there is a difference in SIV cases between the chimpanzee subspecies that are physically separated by the Sanaga River, as you can see in Figure 2. Field research has demonstrated that there are no known SIV positive cases found in the northern purple population, and in contrast, there are known SIV positive cases in the southern orange population. Furthermore, the theory is that P. ellioti may have developed immunity to SIV infection. The research question then becomes, does SIV pathogen resistance contribute to the genetic diversification of wild chimpanzee subspecies in Cameroon? And if so, how? Regarding the method, if we continue to study the single nucleotide polymorphisms, or SNPs, of wild chimpanzees in Cameroon, we may be able to identify the gene that could explain the SIV immunity and diversification. Researchers have already gathered 83 chimpanzees and extracted around 10,000 SNPs from chimpanzee fecal and hair samples. After some analyses, I was given a list containing the outlier SNPs, or significant genetic variations, and the related genes. I searched the genes in NCBI for important publications and research regarding the function of the gene. This gene search led me to finding many genes involved in immune response to viruses like HIV in humans. So I searched each of these genes in a KEGG database to understand the function and utilities in the biological system. And as you can see in Figure 3, I found that interleukin-16 is involved in a family of cytokines that secrete T cells and is involved in a signaling pathway that increases the production of specific CD4 T cells that activate an immune response. And in Figure 4, you can see I found that NCAM1 is involved in a signaling pathway for CD4 as well, and also MHC, which acts as identity tags for harmful antigens. This being said, individuals with a significant variant near this gene may have stronger protection against SIV infection. So I moved on to utilizing our programming to perform spatial analyses. I coded a plot displaying the frequencies of the SNPs in different populations, and as you can see in these maps, there is an obvious variation in the SNP frequencies for interleukin-16 and NCAM1 between the two subspecies of chimpanzees. The red dots show low frequencies of the SNP in the southern population, while the purple and blue dots show high frequencies of the SNP in the northern population. I then utilized linear regression modeling to analyze the relationship between SNP frequency and the environmental variable associated with the SNP. For interleukin-16, it was temperature seasonality, and I came out with a very small p-value. However, because my project focuses on the SIV cases between physically separated chimpanzee subspecies, I focused on analyzing the relationships between SNP frequency and latitudinal area. It came out with a very small p-value for NCAM1, which shows a strong correlation. So my results from my linear regression model support that there is a relationship between SNP frequency and environmental local adaptation. Therefore, my findings can be a contribution to the explanation of the difference and absence of SIV infection between the chimpanzee populations. The SIV immunity could be possible for the P. ellioti subspecies, and that it could be driven by natural selection. With additional research, we can confirm these findings by investigating more than just the single nucleotide polymorphisms, like sequencing the whole gene and its role in the chimpanzees. We can also apply these findings to conservation initiatives along with treatment for HIV in humans. Thank you, and that is my presentation. Have a nice rest of your day.